Hi everyone, how you all doing? Long day? Who was here in my session at 9 a.m. this morning? Hardcore. Who did the meditation session? Is everyone feeling a bit like, whoa? Okay, so hopefully we'll perk you up a bit, because this is a very, very exciting session. The title of the session is The Genesis Machine. Now, I don't know if you were expecting this was going to be some kind of talk about like an AI robot reconstruction of the band Genesis. That's actually tomorrow morning on the main stage, so there you go. But this is, in fact, a fascinating discussion about the implications of the revolution that we've seen over recent years in our ability to read, edit, and even write DNA with a scale and a speed and a precision that, you know, when I was doing my PhD, I could have only dreamed of. And then you add advances in technology and computing into the mix with that, and you have something with truly incredible world-changing potential. And these new technologies, they offer us unprecedented control, that's unprecedented again for anyone doing the bingo, unprecedented control over the genetic code that underpins life, or even the opportunity to create entirely new life forms at our whim. So what can we do? What should we do? And who gets to decide? So I am joined by two fabulous panelists who have spent their lives on the cutting edge of all this biology stuff, trying to figure it all out. So we're joined by Jane Metcalfe. She is the CEO and founder of NEO.LIFE. She's an entrepreneur, publisher, speaker, advisor, manager, and investor, co-founder of Wired, which is a brilliant publication to have been at the heart of. And in 2017, she founded NEO.LIFE, which explores the tools of engineering computer science to alter our own biology. And her book, NEO.LIFE, 25 Visions for the Future of Our Species, is out now. You can find it in the online bookshop. And we're also joined by Andrew Hessel. He's the co-founder and chairman of the Centre of Excellence for Engineering Biology and Genome Project Write, which is about how we write to DNA rather than just reading it. He's a scientist, a communicator, and an investor exploring the future of biology and biotechnology. His new book, The Genesis Machine, Our Quest to Rewrite Life in the Age of Synthetic Biology, that's co-authored with Amy Webb, is also out now, and it is an absolutely fantastic read. So hopefully this session will whet your appetite to go and read it. Now, we're going to have 30 minutes of a quick chit-chat, and then we're going to have about 15 minutes for questions. So I thought I would start with Andrew. If you could explain what this kind of like, what do we mean, or what do you mean by the Genesis machine and all this kind of, you know, biology, technology stuff? How do you see it?
Hi, first of all, I'll just check to make sure that you can actually hear me.
I gotcha.
Terrific. I define myself as a synthetic biologist, and the field of synthetic biology has been emerging really since the beginning of the genomics revolution. It was very hands-on at the start, but really as the technology started to improve, it started to digitize. So we're seeing the appearance of digital biology. Synthetic biology is the intersection of three really crucial technologies. One of them is life science itself, or biology. One is computation and our ability to take digital information and manipulate it. And the other one is automation and robotics. And at the intersection of that, it just comes together to create something we've never had before, which is the ability to read and analyze and now program living machines at scale.
Jane, your publication is called NEO.LIFE. I hope, is Jane still there? Yep, here we've still got Jane. This is a hybrid panel. Please bear with us with any kind of technical glitches. So, you know, you and I have chatted before about this idea. I think with biology, we either see it as healthcare, or, you know, David Attenborough hears pandas and elephants and dinosaurs. So in your view, you know, what is this kind of this new biology, this new life science? How can we think about it? What excites you about it?
Well, first of all, Kat, it's delightful to see you in person, because I know we've communicated mostly via email. So, and we loved your article for the sex cells of lives, which was excellent in NEO.LIFE. Highly recommended reading for anyone who wants to check that out. But yeah, I mean, I think if you're involved in the healthcare system because you or someone you love has a disease or a condition, you know, you would be following the leading edge, perhaps, of developments. You know, on the other side, if you are getting ready to have a baby or taking care of an aging parent or something like that, you might also be engaged. So all of those reasons to keep track are valid, but there's a lot of reasons why even healthy young people should be interested in what's happening, and that's because it's so amazing. The powers that we have right now to transform life are extraordinary, and it's about preventing disease. It's about pushing back the onset of disease, but it's about advanced reproductive technologies. It's how are we going to freeze our eggs? Are we going to freeze our embryos? Are we going to do genetic screening on our embryos before we implant them? If so, are we going to eliminate people with the propensity for mental illness? And if so, how does that change our society? So I just started asking those types of questions and realizing that we have the most powerful tools available to us than ever before, and we will use them to transform our own biology. And you are your own chief medical officer. If you have ever been through the healthcare system, you know that only you knows what's going on in your body and can advocate. And I really feel like it's up to each one of us to really understand our own biology, understand the technologies that are available to us, and to be able to make some intelligent choices, not only for ourselves and our families, but for society at large, because a lot of these capabilities are unleashing just unimaginably complex ethical issues to consider.
Absolutely. Well, let's start with some of the cool stuff. So I want to know, Andrew, in your book, you pull out lots and lots of different examples of how these new genetic technologies, synthetic biology, making new genes, sticking bits of genes into different organisms. What's your favorite application or idea of what we can do with this kind of technology?
Well, my application and my interests are probably different than most people's, but I can tell you where this is really starting to make an impact today for the average person, and it's food. Food is the biology that touches our lives multiple times a day. It's absolutely essential for our fuel and health. It's also such a big part of our social world, and we know that as we continue to grow and thrive, we need to start modifying our food systems for sustainability. So synthetic biology and cellular agriculture are starting to make a real impact in the entire food chain. My particular interest is I like full genome synthesis, which is the ability to take the genome of an organism that has been sequenced, and we have essentially the entire genetic code, and print it from scratch, synthesize it from scratch, assemble that genome using a digital file as the template, and then boot up that genome. This, to me, is one of the most powerful technologies we've ever developed. It's still in its infancy, but it gives us full programming control over an organism.
I did like one of the ideas that you pulled out in the book was the idea that, say, once we are exploring other planets, other moons, and if we were to find some form of life out there, probably a bacteria, I reckon, you could sequence it there. You could then beam the genetic code back to Earth and then recreate that bacteria. I mean, that's pretty cool.
It is. It's literally teleportation. It's been described as faxing an organism from one location to another, and it's being used here on Earth today. The most recent example that people are aware of is just the virus SARS-CoV-2 that causes COVID. It was first identified in China. It was sequenced in China. The code was released, and vaccine development began before anyone had received an actual physical sample of the virus.
So, Jane, for you, what are some of the most exciting applications of this kind of new synthesis of biology and technology that you've seen? Because you must get so many coming across your desk.
Oh, I do, and that's like the great joy of my life. But I spent a certain amount of time as a chocolate maker, which is kind of a funny thing to be able to say when you're talking about science and technology. But the opportunity we have, as Andrew was saying, to transform our food system is one that I actually think about deeply. And I am looking forward to a time when we don't put huge amounts of inputs, water and fertilizer and the air and land and pesticides and all of those kinds of feed to cattle, to monoculture agriculture, to a time when we can actually grow things locally, perhaps in a vat on the roof of your apartment building, if not on your balcony itself or in your own kitchen, and that we can get our nutritional needs met in ways that don't require paving over the roads, shipping things long distance, and having a war in Ukraine cause famine in Africa. I'm looking at not just the opportunity to ferment things in a vat as a way to form new food things to eat, but also a way to reduce the amount of animal suffering and the amount of just energy that goes into all of those things. So the transformation of our food system is clearly on my mind. The other topic, we published a story last week, the week before, on actually canceling menopause. So the idea that women start aging rapidly, it's an accelerated aging process from the time their ovaries stop producing eggs, and it makes them vulnerable to heart disease, mental illness, neurodegeneration. So the idea that we could actually prevent the ovaries from stopping, which means we could potentially prolong the time that women have a healthy later life, is just a mind-blowing capability that some friends of ours are working on. And there's incredibly interesting technology around that could really start to tinker with our notions of, healthy aging, basically.
Yeah, it's an incredibly interesting concept, the idea that we can use these technologies to, turn the clock back. Although I think, Andrew, in your book, you do raise what this scenario could be like if there's a whole cohort of people who are living to completely healthily to 100. This stuff, not just from the cool science of it, but actually will have societal impact.
It's hard to imagine all the societal impacts if we start to live healthy to 100 or 150. But it's not something that's going to happen overnight, obviously. It'll be a shift in demographics. So it might seem weird to us today if we're doing a scenario looking into the future, but I think it would actually feel pretty normal by the time we got there, with just a gradual change.
So one thing I do want to...
It's happening right?
Sorry.
We're already living longer.
Yeah, that is true. One thing I do want to pick up on is this concept. We talk about biology, the fusion of biology and computing, biology as technology. But I wondered if we could muse a bit on how much does the metaphor of DNA as computer code and the cell as computer hold up? Because biology is kind of squishy and doesn't always do what we want. I don't know, Jane, if you want to start with that one.
Yes, well, the whole idea that there is an alphabet that spells out your entire life is just flawed. It is a language, and it changes, it mutates, and some things are interpreted in certain ways and not in others. The idea is flawed to the extent that we take it literally. So but it is code, and it does set us up for things. So how we express that, the exposome, the way we eat, the way we live our lives, that's really the expression that makes us who we are.
And Andrew, you're obviously a big fan. You've already talked about the idea that we can download the code and then boot up a whole new organism. How far do you in your mind think that this metaphor of DNA as code and cell as computer holds up?
It holds up really well at the level of microbes, single-celled organisms. Like a bacterium is essentially clonal. The code is relatively compact, and having bit-level control over it really allows us to learn how to program that organism well, even if it's squishy. Where it breaks down is that we tend to project that onto human and more complex creatures. And you need to appreciate a human is made up of about 50 trillion cells. So it's a much more complicated network. It may still be created and developed from a single program, a single cell, but it becomes complex on many more levels than just the machinery operating in a single cell. It becomes more like the internet, and we know, from the internet that it doesn't respond in the same way as a program because there's just so many more factors going into it and dynamics.
And we are starting to see, though, even though biology is difficult and humans are annoyingly squishy and and hard to predict, we are starting to see this kind of technology coming through to the clinic. I saw some results the other day from a trial of CRISPR therapy for sickle cell anemia and for thalassemia, these blood disorders, with really transformational results. And we've agreed, we seem to have agreed as a society that that's okay to do. Given also the shenanigans that came out around the announcement by He Jiankui a couple of years ago that he had actually, genetically modified human embryos, the outcry of that seems like, okay, that's something as society globally, maybe we think we shouldn't do. So I'm interested to explore where both of you feel like the boundaries are of like, what should we do and what shouldn't we do with this new kind of technology? And Andrew, do you want to kick us off with that one?
Yeah, I think we're seeing these technologies come into the clinic in more and more ways, as you've just pointed out. What's probably more dramatic in the short term is just the ability to diagnose disease using DNA sequencing or nucleic acid sequencing. This has become really a powerful technology, not at the level of the genome, but at the level of understanding what genes are turned on or off, even in individual cells. So the turnaround time for a full human genome now is sub 6 hours at the forefront. That's coupled with machine learning and AI-based analysis tools that is completely changing some diagnostics of rare diseases and even some cancers. So we're definitely seeing this move into the clinic. It will continue to advance in the clinic as we start to have the ability to write code faster. We'll be able to make therapies not just for single gene defects, but to be able to make them dynamically on the fly for individual cancers or individual microbial infections or individual viral infections. As for this goes and what the limit should be, it's really hard to say at this point because we're at the start of this, and we don't want to completely quench the explorations. I think when it comes to human genome editing or genetic surgery, if it was used to actually produce a baby that could not otherwise have been born because of some genetic defect that was serious, I think people would have responded to that better than with the Chinese gene editing report, where it was actually an enhancement. It was not a life-saving genetic surgery. It was a life-enhancing genetic surgery. So I don't think we've seen the compelling case there that will really wake people up to the power of this, to bring people into the world that could not otherwise have been born.
And Jane, in your view, are there things, where do you think we should draw the line with the use of these new technologies?
So there's a very interesting rule that exists among biologists working with genetic engineering, and that is that they can experiment with creating life from scratch or putting together chimeras, in other words, different species together in one life form, as long as they don't let it live more than 14 days. So there is this limit right now of 14 days, which is written about all the time. The scientists say, we did this amazing thing, and at 14 days, we terminated the life. It's a really interesting, completely random sort of number, 14 days. What if it were 21? What if it were 2 months? So that's one of those sliders that we should be thinking about, because it is through this kind of experimentation and this kind of engineering that we learn. We learn how life forms. We learn when it goes wrong. We learn when something gets baked into the code that's going to cause a problem later. So I think that's a thing that might be under reconsideration soon. And I think it's going to alarm people when the conversation reaches, the public awareness, because, at 2 weeks, it's like, okay, fine, there's no consciousness there, there's no pain there, there's no, but is there? And, we may find that pain exists from the very moment of consciousness. I mean, there's no nerves, but, or we may find that it doesn't start until later. So there's huge implications for not only science, but also, the right to life movement and so forth. So I do think that's an interesting one that's going to come up soon.
And kind of moving from the idea of, engineering humans to a bit of the wider world and the risks. Now, I did my science communication training in the 90s, and we were very, very worried that there was this narrative about grey goo. This was the idea that nanotechnology was somehow going to develop this goo that was going to take over the world. And as far as I'm aware, that hasn't happened. I don't know. I think we're good now. And so, what are the risks of this? I don't know if anyone's seen the latest James Bond film, but there's a plot point there that hangs on using genetic engineering, let's put it like that. What really are the risks? And are they realistic, or is this kind of grey goo Mark II that everyone's panicking about and just getting their knickers in a twist? Andrew, do you want to pick that up?
Yeah, in fact, I rewatched the James Bond movie this weekend. And although it's dramatic, the idea of making personalized biological weapons is not far-fetched. We essentially do that whenever we develop a new cancer therapy. We're targeting broken cells within an individual. That level of precision and targeting is astounding, and would certainly apply if we wanted to target an individual in society. So it's not. So in general, I've worried about viruses because, as we've learned the last couple of years, a single virus can be pretty disruptive to our civilization, and we really weren't taking the virus seriously in terms of investing in detection and countermeasures. I hope we don't make that mistake again, but it's not looking good right now, in the sense that we're still treating a pandemic as just a health problem, an epidemiology problem. When I think, given our growing ability to synthesize these agents, we should really be thinking of it more holistically as kind of an immune system for the planet.
Jane, do you want to chip in there?
Yeah, one of the things that I find super challenging is, with my background at Wired, I'm very much a proponent of this sort of do-it-yourself and find your own way, and people can teach themselves and so forth. But I think when it comes to some of this stuff, there are so many ways that somebody who wanted to wish someone ill could use these tools. And it seems to me the best way forward, and we're going to be teaching these, it's postgraduate work, and then it's undergraduate work, and then it's high school work. It's like these tools are available to anyone now. And so controlling access to the reagents and to, gene sequences, the oligos that you would order, there's lots of controls being baked into the systems now, but it's going to be one of those things like security, internet security. You're going to have to be constantly devoting resources to making sure that access to the things that you could use for ill are controlled, regulated, and most importantly, tracked. And so I think transparency is going to be very, very important. Who is working on these tools and what are they doing? And if that's the way we can move forward, I think we can mitigate a lot of the downsides, because I think we'll get very good at identifying people who are doing things that are not in the best interest, that are not following, the generally accepted rules of behavior among scientists or among governments. But I do think we need to sort of chip away a little bit at that sense of, this is mine, I'm going to do it in my lab, I'm not going to have any collaborators, I'm not going to have any oversight. But I do think we also need to keep in mind that it's very hard to create life, very hard to make something live. And the vast majority of science experiments fail, and so the idea that, a giant gray goo is going to take over, I think we're, apart from this idea of being able to generate the killer virus, I think we're a long way away from that, from other aspects of it.
And it does, yeah, it does make me think though, because we're talking about there are now companies out there that will synthesize bits of DNA for you. And as you point out in the book, Andrew, these are run against databases of pathogens to make sure people aren't basically ordering anything naughty. But we now have, we have the dark web, we have people, using cryptocurrencies. And so I'm basically now going to leave this session and go and write a dystopian science fiction novel about people buying virus DNA off the internet. I think we can come back to that. Everyone's writing this down. Don't you dare. That's my idea. I think as well, so, we do need some kind of limitations on this sort of technology. And before we go to questions, I want to bring up the example where there was a moratorium, particularly in the US, placed on using human embryos for research. And actually that was a limitation, but it actually enforced a lot of creativity and probably indirectly led to the development of inducible pluripotent stem cell technology, where you can just take adult cells, treat them with some chemicals, wind the clock back and make them stem cells. Because if we can't get stem cells from embryos, we'll get them from somewhere else then. Screw you. Where are, will limitations open up a sort of a new idea of creativity? Or any comments in that? Either of you who wants to go first?
I think we're going to get through some of the low-level biological risks, because we'll understand that this is a shared problem, really, for our species. We're kind of a monocrop. And when it comes to the other checkpoints and the other, we have to realize that nature is constantly churning a vast number of microbes, bacteria and viruses, et cetera, all the time. And so we just having detection and the growing understanding of biological systems is going to make us more secure in the long run.
Jane, do you have anything to add there before we hit the questions?
Well, I like the fact that Andrew took a general approach to your question, because I'd like to actually go specifically into what you asked about induced pluripotent stem cells, because one of the things that not being able to tinker with human embryos means is, well, okay, even though there are many, many, many human embryos that are currently frozen and might never be used, there's a lot of biological material there that could potentially be used for understanding, again, how to cure disease, how to prevent things from happening, or even to how to enhance humans. But there's something even much simpler, which is the placenta. The placenta is this incredible reservoir of stem cells. And, this biological material gets eliminated with every birth that takes place, at least in this country. I'm not sure what the numbers are in the UK. There's a wealth of stem cells stored in the placenta and in the umbilical cord blood that's just thrown away. It could be used for so many things, and it seems almost criminal. It reminds me of how, in the olden days we used to eat the whole pig or the whole cow, from tip to tail. And, we haven't even brought that level of sophistication, if you will, or basic kind of understanding of the richness of life to our own species. And so I think there's something there.
Thank you.
I think anybody in the audience who's thinking about having children, you should absolutely preserve the placenta, preserve the umbilical cord, cryopreserve it if you can afford it, and tell your MPs that this should be taken on by National Health Service. This should not be a cost to individuals.
Brilliant. Okay, any health policy people in the room? Placentas, please. Thank you. We're going to take some questions now. So if you have a question, please put your hand up. We'll take at the back first.
Okay. My first question would be how the comment at the beginning about working to genetically eliminate mental health issues is different from sort of Nazi eugenics. And also how you can have that train of thought without allowing for how actually technology has really, really altered the mental health build-up of the children that we're raising up now, the next generation that's coming up now. And to then be just looking at, oh, well, how can we just weed out that? I'm just very shocked, to be honest, not shocked.
So, yes, I completely agree. I think the difference between the Nazi eugenics program and this is that, in theory, the Nazis would have had that be a national program. And in this case, it's an individual choice. So individual families choose whichever embryo they want from among the multiple embryos they have. And I grew up with a lot of mental illness in my family. And, you know, had those embryos not been selected, I would never have been born. And, you know, if you look at the extraordinary amount of creativity among people who suffer from mental illness, you know, both in terms of arts, but also science, technology, and so forth, I think it could potentially cripple us as a species to eliminate those people from our gene pool. And yet, that technology exists. And so these are the reasons why we need to understand how to use these technologies. You would certainly want to screen for any kind of physical deformity or anything where you would bring a child into this world who would experience pain. And that's a form of pain. So I think we have to make individual choices, but it's concerning. And I want people to be aware of it. And I want people to talk about it. And I want people to imagine how to do this in a way that preserves diversity. Because that's vital to our species. I mean, if we all choose tall, blonde, athletic, smart people, you know, we'll run into a wall eventually, you know, because that will turn out like a monoculture. That'll turn out to be more vulnerable to threats, less able to evolve as a species.
Thank you.
So and something.
Andrew, have you got anything to come in there on?
You know, I usually stay away from the mental health and genomics because it's a multi-gene trait that we barely understand all the effects of single gene traits on phenotype, how we live. But absolutely, like to give you an example, there's an ethical debate right now with some deaf parents and embryo selection, because that I think really is going to continue to, you know, be one of the lightning rods for these types of ethical discussions. But I don't have an answer for it. Right now I stick really low level with single cells, and am learning by doing and watching with what's happening in the broader perspective of how these technologies are being used in the world.
Thank you.
It's dynamic.
Let's take another question. We'll take one here.
Thank you. Lesser and lesser land will become available for agriculture production, compounded with water shortages. The question is, are there any solutions being offered by cellular agriculture to address those challenges?
I think that's one of the key advantages of cellular agriculture. You don't need a cow to produce milk. You don't necessarily need the whole animal to produce a steak or a lamb chop. So I think these technologies offer the ability to feed the world without having to cover the entire planet in managed farms. It's also I personally don't like the idea of sentient creatures being used as products. But we're again just learning how to do this. We're learning how to do it at scale, at a price point that is cost competitive. But where we're starting to see really exciting examples of this achieving parity. And once we can do that, we can scale the manufacturing in a much more sustainable way.
I will add, you do paint a lovely vignette in the book of like a restaurant critic in 2037, imagining going to like this fancy restaurant and there are cultured meats, but from a much wider range of species. Like, have you tried the axolotl? Or even some woolly mammoth, recreated. So I think there, as well as the idea of like sustainability and actual agriculture to feed the world, I think there's some very cool ideas that you raise. Jane?
Yeah, I was just going to say that, it's one thing to continue to use, extracts from actual animals to create animals without sentience, to just create the animal flesh. But back to your question about sort of design constraints fostering creativity, just the simple idea that we can use alternative proteins, pea shoots, and there's companies that are checking every single like plant compound there is to understand what the various ways they interact with heat or freezing temperatures, what their textures are, what their densities are, what their nutritional values are. So it's fostered this incredible sense of dynamism in the food industry, which I think is incredibly exciting. There's an explosion of creativity and innovation in the food space right now that doesn't involve any animals at all. And, I think that if you can change the way people think about food, you can change the way they think about just about anything. And if we can demonstrate in the food sector how we can use these technologies for good, to have a huge impact on climate change, if nothing else, the amount of greenhouse gases that are associated with agriculture is a staggering amount. And to have everything be local, to have it be maximized nutritionally, because we can tailor the inputs to exactly what the family's needs are. That's incredibly exciting. And I think the food industry is going to be the big win. Food and energy are going to be the big wins for synthetic biology.
Okay, any more questions? Can we take one there, please?
Hi. Earlier in the talk, you mentioned that scientific concepts move progressively from postgraduate research to graduate, and then undergraduates, and eventually high school. So to give you an example of my education, by the time I was in high school, the field of quantum physics was 3/4 of a century old, yet we didn't have anything in curriculum that even mentioned quantum physics. Now, CRISPR is, 10, 15 years old by now? Something
Less
like
than
that?
10, about
Less
9
than
years
10.
old. Yeah.
How can we ensure that new concepts in science, new discoveries like that, make their way into the curriculum of schools and universities so that we can educate people faster on the benefits of these technologies and these discoveries?
I think that's a really important point, and more generally, the public communication about what these technologies are and what we can do. Jane, do you want to take this one first?
Sure. I think it starts with teachers, doesn't it? And we don't compensate our teachers adequately once they've developed these super advanced skills. And it's very difficult to compete with the private sector in terms of the compensation packages. And so we see that with computer science, that it's very difficult to get, great, genius, leading edge computer scientists at the high school level because, especially here in Silicon Valley, there's so many other ways they could use their knowledge. So I do think that it has to do with encouraging our recognition of the value of teachers. But it's also, there's a YouTube video out there that can show you just about everything. So then it becomes a matter of access to, the tools, the reagents, the, whatever else you would need, the specialized equipment you would need. And I think a private enterprise could well spin up opportunities and affordable options for a lot of those things. So it's going to be a mix of, I think, how your government manages education and how important they identify this. I mean, I think teachers with these skills are going to have to be cultivated and recruited and fairly compensated, and it needs to be a priority.
I will wave a little flag for my Genetics Unzipped podcast, because we actually know that a lot of school teachers do use that as a resource for their pupils. It's kind of quite widely used, particularly in A-level biology. Andrew, you are obviously a communicator of this kind of science. How can we make sure that it is getting out there?
It's been getting out there since the beginning. It's just education tends to lag in incorporating some of this material, because they're really trying to see how it fits in and where it'll apply. But I've seen a program called iGEM, International Genetically Engineered Machines, that was booted up at MIT in 2003, now grow into an international program that it encompasses over 45 different countries, and I believe has trained almost 50,000 students now. Learning by doing, all transparent. So there's a growing body of material that any student can access pretty much at every level. The big difference between computers and synthetic biology is, and like just genomics in general, reading, analyzing, and writing DNA, is that the reading and analyzing you can do with a laptop. So the resources are out there, and you can even start to learn by watching YouTube videos and taking online bioinformatics programs. But when it comes to actually doing some of this advanced life science work, we don't have cloud tools that make it accessible. Setting up a home lab in the garage or kitchen is not a viable option for most people. And there are community labs, but there's just a smattering of them around the world. But I've been tracking the appearance of foundries, essentially cloud labs that are programmable, that are making it more accessible. And in fact, in my presentations, I show that my daughter has been making biological art using a cloud lab based in California that allows her to create these really wonderful pictures using many of the same processes and tools that she'd use for a more sophisticated experiment.
Yeah,
so I feel confident. Yeah, I feel confident that this will open up and broaden every year as more people just become aware of the technology and interested in it and see a potential career for it.
Yeah, there's a startup I know about called Arcturus, and they're more for companies and organizations sort of, doing large scale experiments, but it is basically a robotic lab in the middle of nowhere, and you do your thinking on the computer, send them all the things, and like, I want this experiment, I want that experiment, and they will do it with the robots and give it back to you. It's absolutely mind-bending. Unfortunately, we are nearly out of time. So I have one last question for each of you, if we can do this briefly. We've talked about a lot of things, some things very exciting, some things very thought-provoking. I was wondering if there was one particular example that you'd come across that sounds like science fiction, but is actually real. So we'll start with Andrew. If you've got something that you've come across in your research that you're like, oh, wow, that's pretty cool.
Now, it sounds like science fiction, but it's actually real. I think just the idea of being able to boot up an organism from a computer file, most people just aren't aware it can be done. And of course, the teleportation example that you used, which literally is closing the loop from organism to sequence and then back to organism, and really allowing us to beam organisms anywhere on the planet.
Cool. And Jane, you must have come across some pretty wild things in your research.
Well, I do every single day. And so my mind is scanning quickly, like, what am I going to say of all of the examples? I am most stunned by things like optogenetics, the idea that you could actually make a protein fluoresce and then use light to move it around. And they're actually using this to, read and write memories, reactivate memories that have been lost and create new ones that didn't exist before. I mean, it's just in mice, so mice and rats, all the time. But yeah, the fact that at this stage in our evolution, the fact that we can prevent a monogenetic disease in an embryo from manifesting, that's pretty amazing.
Good. Very exciting. Watch this space, I would say. Thank you so much for your attention. I want a huge round of applause for our guests, for Andrew Hessel and for Jane Metcalfe. Thank you very much.